CRISPR is an adaptive RNA-mediated defense mechanism in prokaryote organisms against bacteriophages and other exogenous genetic elements. Using its properties, CRISPR-Cas9 system has been developed as a genetic editing tool. First, Cas9 protein combined with a single guide RNA produces a single double-stranded break in the DNA. Then this damage can be fixed by non-homologous end joining, which often generate random deletions and frameshift mutation, or by homology-directed repair, which can generate precision gene modification using DNA template with partial target homology. The new techniques that have been developed beyond CRISPR-Cas9 have the objective of improving its efficiency. We can facilitate HDR pathway by chemically inactivating the other pathways or by arresting the cell pharmacologically at late S and G2 phases of the cell cycle when most HDR events happen. Other techniques involve conjugation of Cas9. Now we have some examples. In the first, Cas9 conjugation to a part of substrate for a proteasome called geminin can limit Cas9 expression to late S and G2 phases via degradation. In the second, dead Cas9 conjugation to a deaminase allows base editing by the conversion of nucleotides via deamination. The last one, called prime editing, consists of the conjugation of Cas9 with a reverse transcriptase and a prime editing guide RNA. Cas9 nicks one strand of the DNA, the desired edit is inserted via reverse transcription of the RNA template, and the unedited strand is cleaved and repaired to match the other one. Finally, some of the newest technologies in CRISPR are: first, one of the main limitations of CRISPR-Cas9 is the need for a protospacer adjacent motif, or PAM, adjacent to the target. We can create Cas9 variants, making modifications on Cas9 to obtain SPG and then SPYR, which is nearly PAMless. It allows us to edit microRNA, create point mutants, and access polygenes easily to edit multiple genes at the same time. The second one is a tool called Chimera, which is composed by Cas9 and Cas12a. The enzyme Cas12a can generate multiple guide RNA molecules for Cas9, allowing us to edit the genome at multiple locations. It can be used in finding the function of individual exons and paralog genes.